is this the next generation sequencing in forensic science? I just read this book by Kelly Elkins and Cynthia Zeller.
Well, yes, these are my mentors. So they've written a fabulous book, and they taught me next generation sequencing for my research.
You're kidding me. That's amazing. This is great reading during the pandemic.
I know. Nice, easy read. It's perfect.
Can you tell me a little bit about what you did here for your study?
Yeah, absolutely.
I'm gonna move over here.
So my study was on next generation sequencing of mitochondrial DNA, and we were focused on historic skeletal remains that had been recovered in Maryland. So if you look here, I have a photo of the original Calvert town, which is a colony in Maryland.
Okay.
And it's circa 1600s, so our remains are likely from the 1600s, so we think it's one of Maryland's earliest families.
Wow.
And we were presented with a few sets of remains, and we wanted to see, are they related? You know, what's their ancestry look like? So we decided to do mitochondrial DNA, and my research focused on one person who was a 12 to 14-year-old child.
Oh.
And I just wanted to see what their haplogroup was, try to learn a little bit more about their story.
Sure.
So we did mitochondrial DNA sequencing using Verigen's MySeq FGX system, which is beautiful in here. We have a brand new one in our lab.
Oh,
it's awesome.
Congratulations.
Thank you. So, you know, we did this whole experiment from DNA extraction all the way through library prep, and then I used MPA to figure out the person's haplogroup. So this was our results. Of course, with such old remains, we had some, you know, degraded DNA, so it wasn't a perfect profile, but I did get some really interesting peaks for their mitochondrial DNA to figure out their haplogroup. And I entered in all of their SNPs into MPAP, and then figured out the haplogroup.
Oh.
So this 12 to 14-year-old belongs to haplogroup J1C1, most likely.
Okay.
And the most common haplogroups that match that haplogroup are found here in Spain and Portugal, a little bit in Germany, and then Eastern Europe. So it's possible that person came from one of those regions.
That is a fantastic study. So tell me about that mitochondrial DNA. The powerhouse of your cell sticks around longest, and so you get the best DNA from that?
Yeah, so it's really awesome. It is much more resilient than our nuclear DNA, of course. So that's why we focused on it, because we figured that's going to be probably there. We did do CE, and it was terrible. There was no peaks, really. Like, it was not a good sample. So that's why we tried Mito, and especially next generation sequencing, because you just get so much more information. Yeah. And we looked at the control region, so just a certain region.
Sure.
And the cool thing about how you can look at the results is all of this yellow is there's nothing there because it's so degraded.
Yeah.
But any of these blue spots is where it's different from the control sample that they compare against everybody.
Okay.
So there's definitely some differences, which was what allowed us to figure out the haplogroup. So that was really awesome. But yeah, Mito is really cool because we could figure out ancestry. You know, if we had a bunch of samples if we look into some of the other remains, we could try to figure out if they're related maternally and potentially figure out their kinship. So that would be really interesting further on.
Is that a plan for you, Haley, for your group, to take a look at the other remains and see what you've got?
We do want to look at the other remains. We're really excited about it. The new kit that we want to try as well, we want to look at Mito. We're really interested in the new intelligence kit from Verigen.
Okay.
Because we want to be able to upload it to GEDmatch, try to figure out some genetic genealogy work on that, and just hone our skills in that region as well. But we really want to figure out how those folks are related, if at all. Potentially figure out who they were, if they are historically relevant for Maryland, because if they are Calverts, then George Calvert was the first Lord Baltimore. He was the prime person in Maryland, so it would be really cool to figure that stuff out.
Yeah, that is so interesting. And so this is an opportunity that you've had through your university. Is this an undergrad?
So I'm grad, but they do give some opportunities to undergrads too.
I would say,
but I've been really lucky. These fine ladies who wrote this book, they started a lab actually at Towson, which is the Towson Human Remains Identification Lab, Thrill. So we're thrilled to be there.
Cool name.
And we're working with forensic anthropologists to try to identify some of these remains that are uncovered, whether it's through construction or building a road or anything like that. Anytime they come across that, we have the capacity to do next generation sequencing and learn more about those individuals.
That's so fantastic.
Thank you.
I'm wondering, what have you thought about being this student ambassador for ISHI 32? Has it been okay? What's it been like?
It's been fine. No, it's been amazing. I've had so much fun. Just getting to meet people, work with the other student ambassadors, learn about the research they're doing, collaborate, hear from our awesome presenters. I've learned a lot already about genetic genealogy, degraded remains, and so I'm going to hopefully employ some of those techniques in my own research, which I might not have been exposed to had I not listened to them. And then getting to work with all the ProMega folks, they've been so kind to me, and it's just been a really good experience. I'm really glad I was here.
That's so great. Would you recommend then this program to anyone else that you know, or?
Oh, certainly. I would highly recommend it. Yeah, if you're interested in forensic DNA, you should become an ISHI student ambassador, because you're going to have such an awesome experience, learn so much, meet great people, and just gain some new skills.
Yep. Are you looking forward to going to the Disney parks this afternoon?
Oh my gosh, I'm so excited. This is my first time at Disney, ever.
Oh my gosh.
So I want to get my first Disney pin.
Yeah,
like, I got to get the pin.
Visit.
I want to go wear the mouse ears. I want to have the whole experience. I'm very excited.
Absolutely. Well, we're so glad that you came, and thank you so much for
Thanks.
presenting for me.
Thank
That's
you so
great.
much. I
Thanks,
appreciate it.
Haley.